drew a near perfect circle.
That was you last week.
That was me? Cannot be me. It's a beautiful
Was that
circle.
not you drawing the cell?
Okay.
Yeah.
So done. That's it. Right. Can you grab it? Thank you. Okay. So for this chapter, I have to say that actually, if you want to compare cellular respiration and photosynthesis, cellular respiration to me is a tougher of the two. Yeah, it's a tougher of the two. Because cellular respiration comes in many parts. Photosynthesis, just two parts. The light independent, and then, the light dependent, and then there's Kelvin cycle. Yeah? May I draw your attention to the light dependent stage of photosynthesis, which is on page 77? Okay, page 77 of the textbook. Okay, 77 and 78. This is very important because you can draw a lot of similarities to cellular respiration, or differences. Okay, that's a very high level skill to be able to compare and contrast. Okay, let's start with the basics. Let's talk about chloroplast. How many membranes do chloroplasts have? Okay. Okay, we talked about mitochondria first. Mitochondria, how many? Two. Okay, we are very aware about that. Mitochondria, we look at mitochondria, it has the outer membrane and then it has the inner membrane. The chloroplast also has an outer and inner membrane. Okay, so chloroplast has an outer membrane, it has an inner membrane, but it's not quite highly folded like the mitochondria's one. Okay. It has a third layer of membrane. The third layer of membrane is used to form small little M&M steps. We don't consider each one of them as a single membrane, like each M&M not considered one membrane. Okay? Else then we have hundreds and hundreds and thousands of membranes. Okay, we count all of them collectively as one more layer of membrane. Okay? Okay, so collectively, we consider this the third layer of membrane. The one that is used to form all these thylakoids. So we call this the thylakoid membrane. Okay. Some locations. Chloroplast also has intermembrane space. It has this large space over here, the stroma. This is not the matrix, it's the stroma. Okay. And then within all these M&Ms, they are the thylakoid space. Or sometimes they call it the lumen. Okay? It's important that you know all these locations so that you can picture where everything is happening. If you look at page 78 of the textbook, you will see one thylakoid. Okay, one small little M&M. Okay, look at page 78. You can see one M&M. Yeah? So that's just one of them. And you can see a lot of proteins embedded onto it. Okay? So, today, as I run through the light-dependent stage of photosynthesis, I'm going to draw parallels to cellular respiration. Okay, so that you can see the differences and the similarities. Okay, let's first start off with the membrane. Okay, imagine now I'm going to draw the thylakoid membrane. Here's a part of it. Okay, so this is the thylakoid. Okay, I've not completed the thylakoid. I could if I want to. Okay, this one thylakoid. I'm going to simplify drawings from now on. I'm no longer going to draw all the phospholipid bilayer. Okay, I'm just going to represent it by a single line. Okay, too much effort already. Okay, so just going to represent it with a single line. So that's the thylakoid membrane. Let's figure out all the proteins that are present, okay, on the thylakoid membrane. You'll find on the membrane of the thylakoid certain proteins. You'll find some large proteins. They look kind of like this, right? There's like a tube, and then there are like slices that go around it. Yeah? Okay? And then you'll find some proteins along the way. And then you'll find another one of these slice things. Okay? And then you will find ATP synthase. ATP synthase in this case is facing outwards, if I'm not wrong. Okay? So you will find ATP synthase facing this way out. Okay? So here are the players. Yeah? Okay, what do we refer to these as? These are this photosystem 1 and photosystem 2. All these proteins, we can refer to them as electron carriers. Let me orientate you to the location of everything. This is the thylakoid space. Outside of them, therefore, will be the stroma. Okay? If I were to zoom into each one of these photosystems, their role is to harvest light. Okay, let's zoom into one of them over here. You'll find that there's a core. This core we refer to as the reaction. Okay, I'm going to just write reaction center complex. And then you'll find a lot of like segments that surround the reaction center complex. Okay, by right I can draw some more of these segments surrounding it, but I'm not, so that you can see the center. Okay, I can draw more of these ovals surrounding the tube, but I'm not. Okay, just so that you can see the center. Yeah, imagine I peel away a few. We refer to all these as light harvesting complexes. Notice you keep using the word complex. Complex means it consists of many proteins. Okay, it's rather complex. I just draw an oval, but actually it's made up of many proteins. Okay, we refer to them as light harvesting complexes because they come with what you are familiar with since primary school. They come with chlorophyll. I'm using the circles to represent chlorophyll. Each one of these light harvesting complexes contain chlorophyll. Okay, and chlorophyll, as you know since primary school, their role is to absorb light. You will also find chlorophyll in the reaction center. Okay, a special pair of chlorophyll. Okay, a special pair of chlorophyll. Okay, do you also remember I talked about like Kallang waves? How at the end of the day, all of these chlorophyll contain electrons. Okay, they contain electrons. And what's interesting is this: these electrons can be excited. Okay, so what happens is this: when light strikes one of these chlorophyll pigments, the electrons will be excited. Okay, they'll be excited. So you will see in your textbook arrows moving from chlorophyll pigment to chlorophyll pigment. You see that? You see arrows moving from chlorophyll pigment to chlorophyll pigment? There's a misconception. When you see those arrows, students often think that the arrows represent electrons jumping from one chlorophyll to another. It's not electrons that's moving. What is being transferred between chlorophyll and chlorophyll is energy. So what's happening is this: every time light strikes one of these chlorophyll pigments, the energy of the electrons will be excited. The electrons will be excited, it will gain energy. And then it will lose energy. But when it loses the energy, it transfers it to another electron, and the electron gains energy. And then it will lose energy, but as it loses energy, it passes it on to another electron adjacent to it. So it's like a Kallang wave. I'm the electron, I get excited, I raise my hand, I'm excited, and then I get relaxed. But then Anna will get excited, and then she'll raise her hands, and then she'll come back down. We're not transferring electrons. All we are doing is transferring the energy from one chlorophyll to another. Okay? So if you did not include that in your textbook, these arrows represent energy. All they're doing is passing the energy along. I'm passing the energy along from chlorophyll to chlorophyll. And then, think about this. I have so many light harvesting complexes surrounding the one center. All of the energy will be channeled towards the special two chlorophylls in the reaction center complex. Okay? Every single one is channeling the energy to these pair of chlorophyll pigments in the center. And then this is where the magic happens. That's why I call it the reaction center complex. Because at these pair of chlorophyll pigments, the electrons not only get excited, they get so excited that they actually jump out of the chlorophyll. Okay? They are extremely excited. The electrons actually now pass on. Okay, now the electrons move on. These electrons are too excited. They actually pass on to a particular molecule here, which you do not need to know the identity of. We just call it the primary electron acceptor. But that's what we need. We actually needed electrons to kickstart the electron transport chain. Okay, so to summarize, the function of the light harvesting complex is to harvest light. Every time light strikes the chlorophyll pigments, it gains energy, and the energy is passed from chlorophyll to chlorophyll. Everyone channels all the energy to the special pair of chlorophyll pigments in the reaction center complex, and the electrons here will actually be transferred to the primary electron acceptor. Okay? Okay, so now we can continue our story. Imagine now the electron has been transferred to the primary electron acceptor. Okay, we've transferred it to the primary electron acceptor. Okay, and then what happens? This electron will pass down the electron transport chain as usual. Okay, when we talk about electron transport chain, we are transporting electrons down the chain. Not protons, electrons. That's what's going down the chain. So the electrons get transported down the chain. Okay, so that's the first thing we have to take note of. Finally, the electron will go to who? Then we need to have a final electron acceptor. Yeah, maybe I spread it out a bit more. Okay, so over here. Okay, so where will the electrons go? There's always a final electron acceptor for electron transport chain. Okay? We've excited the electron out from the special chlorophyll to the primary electron acceptor and passed down the chain. It needs to be accepted by a final electron acceptor. In photosynthesis, the final electron acceptor is called NADP+. Okay? When it accepts the electrons, it will become NADPH. Okay, so take note, this is the final electron acceptor. Let's compare and contrast. In cellular respiration, what is the final electron acceptor? O2. Remember? O2 is the final electron acceptor in cellular respiration. That's why you need oxygen when you carry out cellular respiration. O2 is the final electron acceptor. Okay? Okay, moving on. Now we have a problem. This special chlorophyll over here no longer contains electrons. Okay, we have a problem here. This special chlorophyll over here does not contain electrons anymore. We need to somehow replace it. Yeah, we need to replace the special chlorophyll, the electrons that's been lost. So we need to top it up. Where can we top it up from? Well, we can top it up from this particular photosystem. So again, there are all these chlorophyll pigments here, the special chlorophyll here, the primary electron acceptor. Every time light strikes one of them, energy will be passed along, energy will be passed along, energy will be passed to the special chlorophyll here. Then the electrons will be excited out, and into the primary electron acceptor, and then it will be passed down the chain, passed down the chain, passed down the chain, and then it replaces the electrons that were lost here. Yeah.
I think they use ATP synthase.
This one, yeah, it's ATP synthase. Yep. Okay, so you see in your textbook there's a lot of additional things like cytochrome complex. You actually don't need to know all those. They are just one of the electron carriers. Okay, they're just one of the electron carriers. So the most important thing you should know is that there's photosystem 1, photosystem 2, a bunch of electron carriers in between, and then there's ATP synthase. Yeah.
When the photon hits, right?
Yeah.
The electrons of the pigment molecule hit.
Yes, correct. Okay. When photons hit the chlorophyll pigment, it is the electrons inside the chlorophyll that gain energy. And then they pass the energy on to the next chlorophyll's electrons. And then they pass the energy on to the next chlorophyll's electrons. They don't pass electrons, they pass the energy. But when everyone passes the energy to this special chlorophyll here, the electrons within there gain so much energy until they jump out. Okay? They jump out and then they go to the primary electron acceptor. But from there, it can now be passed along the chain. Okay, so we have an issue here. This special chlorophyll has just lost its electrons. It needs to be replenished. So somehow we must replenish the electrons here, they are lost, with electrons. Where can we replace? What can we replace it with? So someone needs to donate a fresh electrons to replace whatever is lost here. Okay? We term that thing electron donor. Okay, so there's always a final acceptor, and there's always a donor. Okay? So who's donating the electrons for ETC in photosynthesis? Okay? It's water. Water is the one that's donating the electrons. Then you see in the textbook, it carries out a process called photolysis. Photolysis means use light to break. So we are using the light energy to break the water to get the electrons out. Okay, then we compare to cellular respiration. Who donates the electrons in cellular respiration? Sorry? Oh, go and take. Yeah? In cellular respiration, something comes and carries the electrons to pass it to the chain to start things off. Right? You were here the 2 weeks ago, right? So who gives the electrons? So in cellular respiration, it's NADH, FADH2 that donates the electrons to the electron transport chain. So there are two things you can compare already. We can compare who's the donor, we can compare who's the acceptor. Other things we can compare. Okay? Next, yeah?
The one after the PS1, right?
Yeah.
That's also called electron carrier?
Yeah, electron carrier also. Okay, so we have compared a few things. We compared number one, the acceptor, final electron acceptor. Final electron acceptor. We have compared the the electron donor. What else can we compare? Let's compare where the electron transport chain is embedded. In photosynthesis, it's embedded where? In the thylakoid membrane. Number 3, this is where the electron transport chain is embedded. Where is the ETC embedded for cellular respiration? Inner membrane. Okay, so this is the third thing we can compare. Inner membrane. Okay, next thing we can compare. Same thing like cellular respiration. When electrons are passed down the chain, it uses the energy to pump hydrogen ions. Okay, so let's compare that. Let's compare the direction in which the protons are pumped. Okay, it is pumped from, just now we did the quiz, right? It's pumped from the stroma into the thylakoid space. H plus ions. Okay, stroma to thylakoid space. Okay, this is the fourth thing we can compare, where we pump the ions. What about cellular respiration? Where are we pumping the ions from? And to? So in cellular respiration, we are pumping the ions from the matrix to the intermembrane space. Okay, that's another thing we can compare. Okay, but why do we do all this? So that we can, the usual, set up a proton gradient. And this proton gradient will then, now the protons will then flow through ATP synthase, and then you'll produce ATP as a result. Yeah? So now you okay? Okay, so several things we can compare. Number one, we can compare who's the final electron acceptor, electron donor, where the hydrogen ions are pumped, where the electron transport chain is embedded. What else can we compare? Let's compare the products. What are the products of this electron transport chain? You tell me what are the products?
ATP.
Okay, ATP is one of the products. Anything other products? NADPH. Okay. Now you tell me, what are the products of cellular respiration? So let's compare number 5. What are the products of cellular respiration? Water. Remember? Actually, that's not a product. That is the electron donor. Yeah. But actually, after we donate the electrons, it becomes NAD plus. Yeah. And then FAD. And then you also get water as one of the products. Anything else you get as products? You also get ATP. Right? ATP is also a product. So in both cases, we produce ATP. And you can compare products. Number 5, you can compare products. Anything else to compare? Okay, nothing else to compare.
The H plus ions are being pumped from the stroma to thylakoid space.
Yeah.
Is when the electrons are being transported along the
Yeah.
electron carrier.
Yeah. You try for yourself, right? Are there similarities, are there differences between this electron transport chain and that of cellular respiration? Okay, that's the highest level of notes you can make for yourself. Okay, I'll give you the raw materials to work with. Okay, 5 points you can really compare. The other points you can compare if you really want to. Yes.
Will it be marked wrong if we compare the presence and absence of photosystem?
Okay. Okay, Javier asks, will it be wrong to compare this? Cellular respiration's electron transport chain got no photosystem. Electron transport chain of photosynthesis, light dependent stage, electron transport chain contains photosystem, right? Perhaps, okay, then what's the function of photosystem? What is it? What's the function of photosystem? How will you summarize the function of photosystem? What's it there? Harvest light, is it? Yeah, harvest light. So, in a sense, the electron transport chain of the chloroplast requires light, whereas the other one does not require light. Therefore, there is photosystem, and therefore there is no photosystem. So we go to the root of it all. One requires light. That's why you have photosystem. Anything else you could compare between the ETC of this and the ETC of cellular respiration? Anything else to compare? What's similar to you? Any similarities?
Product.
Oh, also, okay, what's the similarity in product?
ATP.
Okay, ATP is produced. Okay, that's a similarity. Any other similarities?
Presence of ATP synthase.
Presence of ATP synthase, both of them have, right? Okay, so that's a similarity. Any other similarities?
Proton gradient must be established.
You need proton gradient for ATP synthase to work. Similarity? Similarity. Okay? So you can train yourself this way. Right? Find the similarities, find the differences. The biggest difference of this electron transport chain is truly that it has these photosystems to harvest light, to get the electrons moving in the first place. Okay? So the best thing you could do is for yourself, when you make notes, is to draw a table of comparison. That's really the best thing you can do for this chapter. Okay? If you have not made a table, maybe you want to try. Okay? This is the best thing you can do for yourself to really consolidate everything together. Okay? It's unwise to study photosynthesis by itself. Study it with cellular respiration, and you are good to go. It is one topic together. Kavi's question is very good. He ever say, how is the glucose? Yeah, Kavish ever came up with a question that links everything together. How is the glucose induced? No.
How the glucose form?
Because plants have both mitochondria and chloroplasts. They have both. So I think his question was, how is glucose formed and used in plants? That is crazy, you know. How is it formed? You have to describe photosynthesis. How is it used? You have to describe cellular respiration. I think it's an awesome question. I'll use it for next year.
Next year, not for us.
No, why?
Yeah, it's like okay, I'd like to show you can find all this again. Okay, it's not. Okay, I have to say, your biology textbook is dead. It's not alive, you know. It's like, it's dry. Okay, that's the reality of it. It is dry because it's just pictures. Where can you supplement? Really, go to this site. Find videos to animate whatever you are reading. So, if you want on photosynthesis, you can go and search for photosynthesis here.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. The energy most age. Plants appear green because they're also known as photoautotrophs. The chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Kelvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Kelvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called Atp Synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH.
Okay, then the next part of photosynthesis is the Kelvin cycle. The Kelvin cycle, remember, Anna, was it you helped me record in class? Yeah? So the details of it, truly, you don't need to know the details per se. Right? What's important for you for the Kelvin cycle is to know what is required for it, which is NADPH and ATP. And then you need to know the products of it, which is G3P. And 2G3P become glucose. So I think the rest of this video will show that.
When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Kelvin cycle reactions. The Kelvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Kelvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.
Okay, I'm going to forward the video of me teaching it again to our classes, and you can access it directly. don't have to place too much emphasis on the Kelvin cycle. Yeah, don't need to memorize all the steps. Okay, it's not necessary. Right? What's more important is you just have an understanding of what's produced, what is needed to get it running. Okay, what's needed to get it running? Whatever the products are of the ETC. Okay, so again, for this chapter, really, I don't see photosynthesis and cellular respiration as separate. They are one and together. Okay, so to study them well, please make some sort of a table to link them together. The more you link, the better prepared you'll be for this chapter. Okay, go and compare and contrast. Okay, help yourself out for that. Okay, we've come across such questions before also in your quiz. Okay, so really, a lot of learning can be done by just learning from your mistakes. I'm not sure if it's been said. Before 3 should have the and now you can, do you have questions you want to ask? Or you just re-forward the class, since you sent it before actually. Yeah, you can just forward the class. You just forward the class again. I think in 4.4 I've also sent a video before. I felt like I did that.
No.
Any questions from the floor?
Three phosphoglycerate is
3-phosphoglycerate is what? No, it's different. G3P is glyceraldehyde 3. Yeah. Oh, why twice is it? Okay. So each cycle that you go through, you only produce one G3. And then G3P is made out of 3 carbons. And glucose made out of how many carbons? 6. So that's why you run out of points. Then you can get correct, they're not the same. There are a lot of reactions that occur along the way. Yeah, correct. So it's very easy. So you can see here together a series of reactions involving ADP. You wanna test to see if you made any improvement? Have you all stand down yet? No. Still going on.